Hello friends, my name is Brajendra. As you all know that diagrams are very much essential in biology. Okay, so if your diagram is correct, you can score good marks in biology. Okay. So today I will teach you how to draw good diagrams. The first diagram which I am going to draw now is a very important diagram which usually asked in examination. The diagram is the physical structure of DNA. Okay, for drawing this diagram, you have to take two pencils at one time. Okay. So we have to draw 4 nitrogen bases. Okay, in each complete loop. A double bond T, C triple bond G, G triple bond C, T, A, C, T. Okay, and where the loop is incomplete, then you can draw a 2, 3 or 1 nitrogen base pair also. Okay, so as you can see over here, this angle is smaller, this is a bigger angle. So this is called minor groove and it is called major groove. Okay, so the distance is 34 angstrom from here, and distance between two nitrogen bases is 3.4 angstrom, and distance between the two strands is 20 angstrom. Okay, so this is the physical structure of DNA. Diagram, the written diagram is 2 marks, in which the half mark is for sketch and 1.5 marks for labeling. Okay, this is the first diagram which we have drawn. Okay, fine. Okay, the next diagram which I am going to draw is a very important diagram, which is usually asked in examination. Diagram name is anatropous ovule. The weightage of this diagram is 3 marks. So how you draw the diagram? Okay. The next part is the labeling part. For labeling, the lines will be straight. The arrow should point towards the diagram and not towards label. So this diagram is of 3 marks weightage, in which 1 mark is for drawing and 2 marks is for labeling. So for 2 marks labeling, you have to label any 4 labels. You have to make any 4 labels. Okay. And after completing a diagram, so that you should write the name of diagram below. And after finishing a diagram, you have to make an outline from outside. Okay, there are certain rules for drawing diagram. The first and foremost, very important rule is that your diagram should be proportionate. It should not be too large, it should not be too small. It should be proportionate, means this diagram should be about almost of 10 to 12 lines, not more than that. Second thing, the labeling should towards one side, either left hand side or right hand side. See, in exam you need to label any, you need to make label any 4 labels. So try to label any one side, either left hand or right hand side, where you are comfortable, but don't make it clockwise. Then last thing, your arrow should be towards the diagram, not to a label. Okay, these are the rules of drawing diagram. And last one, the outline you should draw. Okay.
Please start.
Okay, the next diagram which I am going to draw now is a very important cycle. Okay, cycle. The name of cycle is called as C4 pathway. C4 pathway. It's also called as HSK pathway. The full form of HSK is hash, slag and Korsack pathway. The video diagram is 3 marks. So how you draw the diagram? Okay, so there are two types of chloroplast, mesophil and bundle sheath chloroplast. The carbon dioxide enter inside the which chloroplast? Mesophil chloroplast, and it combines with PEPA. It's a 3-carbon compound and forms OAA. It's a 4-carbon compound. 3 carbon plus 1 carbon forms what? How many carbon? 4 carbon. This OAA undergoes reduction with the help of NADPH2 to form malic acid. Now this malic acid, it get transformed from which chloroplast? Mesophil chloroplast to bundle chloroplast through plasmodesmata. Okay, so after it get transferred to bundle chloroplast, this malic acid undergoes decarboxylation. CO2 is removed outside, and this CO2 get combines with RUDP undergoes which cycle? Calvin cycle, and forms carbon dioxide from malic acid. This malic acid can convert to form pyruvic acid. So 4 carbon after release of 1 carbon from common carbon 3 carbon. Now this pyruvic acid this pyruvic acid get transferred from bundle chloroplast to mesophyll chloroplast through plasmodesmata, and after entering inside this mesophyll chloroplast, it undergoes phosphorylation with help of ATP to form PEP again, and formation of PEPA again continue the cycle. Okay, thank you. So this diagram is of 3 marks. Half mark for every step. Means if you have written PEPA, you get half marks. You get OA, you get half marks. So the marks you get for half marks for each every steps. Okay, fine. The next diagram which I am going to draw it now is called as double fertilization. Again, the weightage is for 3 marks. So how will you draw the diagram? the pollen tube enter inside the ovule through micropyle. So this entry of pollen tube inside the ovule through micropyle is called porogamy. So after entering inside the ovule, this pollen tube it gets stuck to any one synergids. So inside the synergids there are filiform apparatus which attracts the pollen tube towards itself. So the pollen tube get fused with the any one synergids and try to absorb all its nutrients. So after absorbing nutrients, the mucilage, the pollen tube, deeper pollen tube will get swelled and will get burst open to release the male gametes. The first male gamete get fused with the synergids or with the egg cell. Okay, the first male gamete fused with the egg cell. So this fusion of the first male gamete with the egg cell is called syngamy. And the second male gamete fused with the secondary nucleus. So this fusion of the second male gamete secondary nucleus is called triple fusion. And lastly, I draw the outline. Okay, thank you.